When this image appeared on millions of screens this week, the reaction was immediate. Many viewers thought they were looking at a man who had been burned, or attacked, or worse. I take it orally, but it's a nasty drug. It causes crazy stuff like my body can't grow skin, and so I bleed all out of a whole bunch of parts of me that shouldn't be bleeding. The man speaking is Ben Sasse, former U.S. Senator, father of 3, and stage 4 pancreatic cancer patient. And the drug causing that face? It may be the most important cancer breakthrough in a generation. Daraxonrasib, the molecular glue that's fighting one of cancer's most dangerous villains. In December 2025, Ben Sasse announced the news on X with characteristic bluntness. He had been training for triathlons. He'd had persistent back pain. Then came the scans, and a doctor's verdict that cut through everything. Here's a hard fact. Ben Sasse's torso is chock full of tumors. Stage 4, already metastasized, not operable. 3 to 4 months to live. The cancer had spread beyond the pancreas. Lymphoma, vascular cancer, lung cancer, liver cancer. 5 forms of malignancy, all originating from one corrupted molecular signal. But Sasse did not simply accept the prognosis. He enrolled in a clinical trial at MD Anderson Cancer Center in Houston for an experimental drug called Daraxonrasib. I'm at day 99 or something since then, and I'm doing a heck of a lot better than I was doing at Christmas. To understand why Daraxonrasib is such a big deal, you need to meet the villain. Deep inside nearly every cell in your body is a protein called rash. It's a molecular on-off switch. When activated, it sends a signal: grow, divide, survive. Normally, an enzyme flips it back off within seconds. But in about a third of all human cancers, a mutation permanently jams the switch in the on position. In pancreatic cancer, roughly 90% of tumors carry a RAS mutation. For 40 years, scientists knew this was the engine of the disease. The problem? RAS had no docking pocket for a drug to grab. It was smooth, featureless, undruggable, they called it. Scientists at Revolution Medicines asked, what if we don't try to grab RAS directly? What if we use another protein to do it for us? The answer came from nature itself, a molecule called sangliferin A, produced by soil bacteria. It had one remarkable property. It bound tightly to a human chaperone protein called cyclophilin A, or CyPA. Revolution Medicines transformed sangliferin A into Daraxonrasib, a molecular glue. Step 1. Daraxonrasib binds to CyPA, reshaping its surface. Step 2. That newly shaped CyPA surface fits precisely onto the active GTP-locked RAS protein, forming a 3-way complex. Step 3. The complex physically blocks the surface of RAS that downstream proteins like BRF use to receive the grow signal. The highway is closed. The tumor's instruction to proliferate goes silent. The early numbers are historic. In phase 1 trials, nearly 90% of pancreatic cancer patients had their disease stabilized. Patients survived a median of 13 to 16 months, nearly double the standard chemotherapy outcome. In lung cancer trials, 38% of previously treated patients saw their tumors shrink, with responses lasting over 15 months. The FDA has placed Daraxonrasib on a fast-track review pathway. Phase 3 results, comparing the drug against chemotherapy, are imminent. But there is a price. Because RAS signaling is also essential for normal skin cell regeneration, the drug disrupts the epidermis throughout the body. Skin can't renew itself fast enough. It becomes fragile, raw, bleeding, especially on exposed areas like the face. His face that shocked millions is not a wound from the outside world. It is the collateral signature of a drug fighting a war inside his body. Cancer sucks. But I'm pretty grateful that cancer is a stake against my delusional self-idolatry. Because RAS mutations drive a third of all human cancers, Daraxonrasib is potentially a platform for colorectal cancer, melanoma, and beyond. This molecule is poised to unlock the most stubborn door in oncology. The door that has been locked since 1982. Death is terrible. We should never sugarcoat it. But it is great that death can be called the final enemy. And then there will be no more tears. In the meantime, a bacterium from the soil, a chaperone protein, and 40 years of failed attempts have combined to give one man, and perhaps thousands more, a little more time. For more medical stories, visit our website, unbedsidemanners.com.